So we start out with the initial strand of DNA. Helicase acts on the strand, unwinding it to create what is called a replication fork. Because the DNA is in a helix shape, if it were to be unwinded, it would supercoil. That's where gyrase comes in. It prevents supercoiling and allows the DNA to replicate in an orderly, efficient manner.
In order to start replication, an RNA primer attaches to each strand. Because the strands are antiparallel and run in opposite directions, one strand, called the leading strand, will replicate down the replication fork continuously, while the other strand, called the lagging strand, will replicate discontinuously. After the RNA primer is formed, DNA polymerase III adds on nucleotides to the 3' end of the RNA primers and helps to create a chain of nucleotides.
On the leading strand, DNA nucleotides attach to the original strand and a chain of nucleotides continuously forms down the whole strand. On the lagging strand, an RNA primer is added after a few bases each time on the strand.
After replication is complete, DNA polymerase I removes the RNA primers from the newly formed strands. Then, ligase fills the gaps created by the DNA polymerase I, thus replacing the RNA primer with a DNA nucleotide. And there you have it, DNA replication.